Dear all, welcome to this video. Please hit the subscribe button so that you automatically get updates for upcoming videos and lectures. Thank you. Enjoy watching.
So we'll start with introduction first. Fungi are classified into eukaryotes. As you all know, the difference between prokaryotes and eukaryotes. Eukaryotes are the cells which have defined nucleus. They are non-motile. They cannot move from one place to another. All fungi are chemoheterotrophs. Chemoheterotrophs means they require organic compounds for energy and carbon to grow and for their nutrition. Now these can be aerobic or facultative anaerobic. That is, they can grow in presence of oxygen or even in the absence of oxygen, and they can be morphologically divided into two classes, yeast and hyphae. What is the difference between them? Yeasts are the unicellular fungi which reproduce asexually by either budding or fission. We have already seen this process. And hyphae are the multi-cellular fungi which reproduce asexually or sexually. The synonym for budding is blastoconidia. Whatever buds are formed, those are also called as blastoconids. So that's why this process is known as, scientifically it is known as blastoconidia. Now most fungi, they occur in hyphae. Hyphae are thread-like structures, branching and thread-like. So here you can see the long threads can be seen. So those are called as hyphae. Now these hyphae can be divided into two, coinocytic hyphae and septate hyphae. Now coinocytic hyphae, you can see that there is no cross walls. What are cross walls? Here you can see that there have been segments separated, these one with the help of walls. So these known walls are known as cross walls. Whereas here you can see there is a single complete unique strand. No cross walls or no compartments can be seen. So these are coinocytic. And these are septate one. And there are clamp connections which connect the two hyphae elements together. Now this is the process of formation of septate hyphae. Initially budding will take place, that is blastoconidia formation. These buds will cut down into two, and on one bud another bud will be formed like this. And here you can see normal small buds have been formed, and these individual buds will start to grow and they will form a pseudo hyphae. Okay, then here you can see the pseudo hyphae has been formed and all the nucleus have been separated. But this is a coinocytic. Coinocytic means what? There are no walls present, no compartments present. So initially it will form a coinocytic hyphae. Later on, the septate hyphae. After the walls have been prepared, then it will form a septate hyphae, and this septate hyphae will further lead to clamp connections. So these are the projections are known as clamp connections, which connect the two compartments together or the two cells together. Now, what is the difference between fungi and bacteria? Fungi are eukaryotic organisms. Bacteria are prokaryotic. The pH required for fungal growth is 4 to 6. The pH required for bacteria is 6.5 to 7.5. So, in short, fungi grows in acidic pH. The optimum temperature required for fungal growth is 25 to 30 degrees Celsius. Here it is 32 to 37 degrees Celsius. Cell wall or cell membrane, it contains sterols, whereas bacteria does not have sterols except in mycoplasma. Oxygen requirement, fungi is strictly aerobic and facultative. Anaerobic, here there is a different classes of aerobic to anaerobic. Complete anaerobic bacteria are also present. Light requirement, now in case of fungi, light is not required because it cannot synthesize its own food. But if a bacteria is photosynthetic, then it can synthesize its own food. So it will require light. Carbon source, organic, here both organic and inorganic can do. Sugar concentration in media for growth is 4 to 5%. Fungi require high sugar content, whereas bacteria don't. It requires only 0.5 to 1%. Cell wall components, there it is usually made up of chitin, cellulose or hemicellulose, whereas bacteria, as you all know, they are made up of peptidoglycans. And the susceptibility to antibiotics, both are susceptible to same kind of not same kind of antibiotics. The fungi are sensitive to cresofulvin, which is an antifungal drug, but resistant to chloramphenicol and penicillin, which are antibacterial drugs. Whereas bacteria is resistant to cresofulvin because cresofulvin is an antifungal drug, and they are sensitive to chloramphenicol and penicillin because those are antibacterial drugs. Now, what are the characteristics of fungi? First of all, heterotrophy, that is, they cannot prepare their own food. So these heterotrophs, as we have discussed in bacterial chapter classification, that the heterotrophs are further divided into saprophytes, symbionts, and parasites. Saprophytes are the bacteria which here these there will be fungi which will feed on dead tissue or organic waste. Symbionts means where both the organisms are benefited, that is fungus as well as another organism, and parasite will be feeding on the living host tissue where the parasite is benefited and the host cell is at loss, and those are usually pathogens. Some parakeets are obligate parasites while others are facultative or non-obligate. Okay, so parasites, some are obligate, means which will require a living host to survive, whereas some will not require a living host in order to survive. They can even grow on non-living organisms. Next is body form. Now, body form, as we have discussed, is unicellular, filamentous, all hyphae, thread-like structures, long tube-like structures, you can say. Mycelium is aggregate of hyphae. Too many hyphae together form a mycelium, and sclerotium is what the sac, hardened mass of mycelium that generally serves as an overwintering stage. Means in winter they go in dormant state and there they store the spores in it, so that in favorable condition these spores will sprout out or they will be spread through and then the growth of fungus will be take place. So the place where the spores are stored, those are known as sclerotium, and these are multicellular such as mycelial cords, rhizome, moths, and fruit bodies, that is mushrooms. So those come under multicellular. So here you can see the sporangium or sclerotium, which contains spores. These are the sporangiophore, which holds the spores, and these are the rhizoids or stolons, which are embedded in the host cell. Next is the fungus is often hidden from view, means you cannot grow it easily. It grows through its food source and excretes the extracellular digestive enzymes and absorbs the dissolved. So externally digestion process takes place and then it will absorb whatever digested material is there. Okay. Next is indeterminate growth. You cannot control the growth of fungus. It is indeterminate unless and until it gets nutrition, it will keep on growing. Spores, they are both sexual as well as asexual. Asexual spores will be they are the product of mitosis, and sexual spores are the product of meiosis. Now, what is the purpose of the spores? Why the spores are used? First of all, it allows the fungus to move to new food source. Obviously, through spores, the area of that particular fungus can grow because it can spread out. So that's why new food source will get. Resistant stage allows the fungus to survive in the periods of adversity. As I told you, in harsh conditions, they will remain in dormant state, and in favorable conditions, they will germinate. And means of introducing new genetic combinations into population. So if it happens that the two spores combine together and they form diploids, so in this way, genetic combinations may take place. Next is vegetative phase of fungus is generally sedentary. Okay, that is dormant one, and cell wall, as we have already seen, it is made up of cellulose or chitin. Food storage, generally in the form of lipids and glycogen, they store the food. As I told you, eukaryotes through nucleus and other organelles present. All fungi require water and oxygen because they are not anaerobic, so obviously they require oxygen. And the fungi can grow in almost every habitat imaginable as long as there is some type of organic matter present and the environment is not too extreme. If the environment is not much extreme and if some organic matter is available as a nutrition for them, then they can grow. And there are about 1.5 million species till now estimated as fungus. Now, classification, if you see, the phylum Thallophyta divided into fungi and algae. Okay, so there are two. What is the difference between fungi and algae? Algae have chlorophyll, so they can synthesize their own food. Fungi don't have chlorophyll. So fungi is again divided into pseudomycetes and eumycetes. And pseudomycetes are divided into schizomycetes and myxomycetes. Schizomycetes are the bacteria and myxomycetes are slime molds. And morphological classification, if you see, then it is molds and fleshy fungi, yeast-like fungi, and dimorphic fungi. Dimorphic fungi are the fungi which show two different morphologies. Okay, so depending upon the conditions, the morphology change and they adapt to the environment. Next is systemic classification of the fungi. So again, if you see a systematic classification, then these are the different classes which are included under fungi. That is Phycomycetes, Ascomycetes, Basidiomycetes, Fungi Imperfecti, and Dermatophytes. Dermatophytes are, they are the fungus which grow on skins, dermal layer. So that's why it is known as dermatophytes. Now, what is the application or importance of fungi? First of all, sources of antibiotics. Many antibiotics are synthesized from fungi like penicillin and all. All those have been obtained from penicillin, notatum and all. Then sources of enzymes. Even different enzymes have been synthesized with the help of fungi. Then production of citric acid, oxalic acid and gluconic acid, which is highly used in food and in other food-like substances. They alter the texture, improve the flavor, increase palatability and digestibility of natural and processed food. So yeast and all you must have heard. So due to which the texture, flavor, everything is can be altered. Then edible varieties, even mushrooms and all, whichever are edible varieties available, we can eat it as a food. Fermentation of beverages and juices, alcohols and all are prepared. Juices are prepared by fermentation process. Industrial alcohol is also prepared by fermentation. Is the alcohol which is used as a beverage, but as well as the industrial alcohol, both are prepared by fermentation process with the help of fungi. Then breakdown of complex organic substrates into simpler ones. As I told you, that they carry out the extracellular digestion process and then they absorb the food. So this extracellular digestion can be utilized for converting the complex organic substrates into the simpler ones. Now this is the reproduction cycle of fungi through spores. Now as you all know that both the sexual and asexual reproduction may take place. So here you can see these are the spores. These are spores on germination will give rise to mycelium. Mycelium will further give rise to spores. Okay, and this cycle will go on. So this is a asexual reproduction. Only one parent is taking part, and number is not increasing. It is remaining the same. And number of chromosomes, that is, it is remaining haploid, that is, and not given. Now, if by chance it happens, now this mycelium, if it fuses with another spore, means two mycelium come together or two spores come together, there is plasmogamy, that is fusion of cytoplasm. So it will form N plus N, that is the dikaryotic stage, and further it will lead to karyogamy, that is the fusion of nuclei, and it will form 2N, that is the diploid stage. And as a result of meiosis, spore producing again structures will be produced, which is not mycelium, but they are given some synonymous name. And after that, spores will be released and they will further germinate, and again they will lead to formation of mycelium. So this cycle where 2N is involved, that is two sets of chromosomes are involved, and it is called as sexual reproduction. And if a single set of chromosomes is involved, then it is the asexual reproduction. Thank you.